Everyone, Brian here, and in today's video I will be going over exercise 5-11, which is the starch hydrolysis test, and exercises 14 and 15, which is the gelatin and DNA hydrolysis test, respectively. Now, in the previous video, I went over the two tests that focused on intracellular enzymes, and the last three tests that you will be performing this week will focus on extracellular hydrolytic enzymes. Of course, just to do a quick review, these enzymes that are called hydrolytic enzymes use water to help them break down large complex molecules. And of course, as with many other tests, testing for enzymatic activity is a great way to be able to differentiate large groups of bacteria. Alright, so let's jump into the first test here. The microbes that you will be handling for the starch hydrolysis test is B. subtilis and E. coli. Of course, once you have your bacterial cultures with you, you'll not then need to grab the rest of the materials that you need, that being your tube rack, inoculating loop, and your Bunsen burner. And finally, of course, you will need 1 starch agar plate and 1 grams iodine, which is usually kept in a brown jar that looks something like that. Okay, so why don't we dive into how to prepare and inoculate your plate. So the first thing you want to do, of course, is separate or flip your plate upside down and draw a straight line across, creating 2 halves. Of course, the first half will be designated to B. subtilis and the other half being designated to E. coli. And once you have your plate entirely labeled and ready, you can then transfer the bacterial culture onto your plate. Of course, first following all the standard aseptic procedures before doing that. So what you want to do is, once you have your bacterial culture, to just perform a simple spot inoculation, making sure that you are using the bacteria that is assigned to that half. Now you'll then repeat that procedure, of course, with the second organism that you are working with. And now for this grams iodine that you see here, you will actually not be using that until the second part of the lab. So why don't we go back into slideshow mode, and I will show you what grams iodine will do. So when you come back, this plate that you see here has already been introduced to grams iodine. The grams iodine will come with a dropper itself, and all you have to do is, of course, cover the entire plate with the grams iodine itself. And once you do that, you will then notice that there is this distinction here between these 2 colonies. And what you are seeing here is that this organism that is positive for starch hydrolysis has released these enzymes known specifically as alpha amylase and oligo-1,6-glucosidase. And these enzymes will break down the starch in the surrounding area. And as you can see here, starch is this long, complex chain of glucose molecules that have been attached to each other. And now the thing is that iodine only interacts with starch itself. And so organisms that can then, of course, break starch down will then sort of remove the starch nearby the surrounding colony, of course turning it into its individual component part known as glucose, which also then is most likely taken up inside the cell to have been used for energy. But iodine, of course, interacts with starch, and it forms this brown color that you see all over the plate. So if you take a look at this results table here, this test is quite simple in terms of what you're looking for. If the organism has this distinct clearing around the colony, then you know that the organism is positive for these enzymes here that can break down starch. While this colony here, as you can tell, there is no clearing around it, is not capable of producing these enzymes, thus is not able to break down starch. All right, so why don't we quickly review, and these are the three concepts that I want you guys to take away from this test. Of course, this test is designed to look for organisms who can break down starch. Those that have that ability is due to the fact that they are able to produce an enzyme that can, of course, break the starch down into glucose. Those that do not, the iodine that you place on the plate will then interact with the starch that is within the test media itself. And of course, you will see that those organisms that are negative do not have that distinctive clearing around it. Finally, more so in terms of application, this test aids in differentiating members from the genus Corynebacterium, Clostridium, Bacillus, Bacteroides, Fusobacterium, and Enterococcus, showing you that this is a fairly good test to use to kind of help break down large groups of bacteria in a very broad manner. Right, so we are now done with starch hydrolysis. Why don't we go over the next test, which is the gelatin hydrolysis test. And the organisms that you will be working with are, once again, B. subtilis and E. coli for this lab. This step here is probably very familiar to you now. You just want to gather all of your materials, with the only difference being the 3 nutrient gelatin stabs that you will need. Okay. So hopefully when you get your test, the bacterial culture is not open like that. But for this test, actually, you will be using a needle because you will be stabbing the nutrient gelatin. Of course, you want to make sure everything is sterile, that your tool that you're using to transfer is sterile as well. And once your needle is ready, you can then use it to just kind of grab some of the culture that is inside. Once you do that, of course, you want to first sterilize your tube, and you can then proceed to stab the test media itself. Trying to go as close to the bottom as you can, but not actually touching it, and then gently pulling the needle out. You will then, of course, repeat this procedure for the next nutrient gelatin stab, and of course leaving this one alone because that is your control. Okay, so why don't we go back into slideshow mode? Oh no. Okay, sorry, we had a little bit of an issue there. Anyways, let's continue on. So when you get your tests back, it should look something like this, where one of them has turned into a liquid while the other still has remained in its solid form. Looking at the results table, we know that when it has turned into a liquid, that the organism is positive for this enzyme here, gelatinase, and thus it has the ability to break down this molecule here known as gelatin, which is this complex protein comprised of very long chains of polypeptides. Now, gelatin in that form, of course, is solid. However, as the bacteria produces gelatinase, and gelatinase then interacts with gelatin, breaking it down, it will then break it down into these long polypeptide chains and then further break it down into the individual amino acids that it is comprised of. And once it has been broken down into those amino acids, it can no longer maintain this solid form, and thus it turns into a liquid. And so we know that this organism here is positive. Now there is one thing to keep in mind here. Sometimes these nutrient stabs will have been placed into a cooler, and it is best not to immediately take the results down when they have just been taken out, as we know that the cold temperature will of course turn the test media itself solid, and so if you do end up making the determination too soon, you can accidentally label an organism negative, assuming that the organism did not have the ability to break down the gelatin itself because it is still somewhat frozen. So please be careful with that. Make sure to let the test tubes warm up to room temperature before actually writing down your results. All right, so let's just quickly go over. The test is designed to look for organisms that can produce this enzyme known as gelatinase. That enzyme, of course, will then interact with the core substrate known as gelatin by breaking it down first into these long polypeptide chains, and then, of course, breaking those further down into the individual amino acids that it is comprised of. And of course, that process will turn the gelatin that was once solid into a liquid. And if that is what you see after incubating, then you know that the organism you are dealing with is positive for this hydrolytic enzyme here. All right, so finally, what is the best application for this test? It is known to be able to separate Staphylococcus aureus from Staphylococcus epidermidis, in that Staph aureus is positive and has the ability to break down gelatin, while epidermidis lacks that ability. So in a hypothetical situation where you are fairly confident that you are dealing with either one of these two organisms, this is a great test to kind of separate those two from each other. In addition, it is also able to separate species within Serratia and Proteus from other members within the large family of Enterobacteriaceae, as those species within those two families are known to be positive, while other members within Enterobacteriaceae are negative. All right, so moving on to our last and final test, the DNA hydrolysis test. In that one, you'll be dealing with three organisms here, Staphylococcus aureus, S. marcescens, and of course E. aerogenes. And Staph aureus being a BSL-2, you will of course need to wear gloves for this lab. All right, so for this one, you can then go back to your inoculating loop. Once you have all of that, you will then need to grab one DNase test agar plate, which has this sort of tealish green color. And we will go into why the plate has that color. But first, I will then demonstrate what we'll have to do when it comes to inoculating your plate. All right, so before we actually go into transferring the organisms onto your plate, what you'll first need to do is flip your plate over onto the bottom side and draw two lines splitting the plate up into 4 quadrants. So try your best to imagine that there is this little cross here separating the plate into 4 little sections. And of course, in the first 3 sections, you will then label with the organisms that you are working with. And of course, those sections are then assigned to those organisms. So please be careful when you are spot inoculating that you are using the correct organism there. And of course, the last quadrant here you will just leave alone, as that will be your control. All right, so once you have your plate labeled, you can then transfer the bacterial culture onto your plate. Of course, making sure that all of your tools are sterile. You will then just grab some of that culture here and perform a simple spot inoculation going across diagonally. And then you will do that 2 more times for the next 2 sections. Okay, so when you guys come back, what you should see is something along the lines of this plate right here. So this natural green color that you see within this plate, well, it's not really natural, but this green color is due to this chemical compound known as methyl green, which plays the role of the indicator in this test. And these long blue lines that you see are the polymerized DNA that is the main substrate for this test itself. And methyl green can only form a complex with polymerized DNA. Now, polymerized DNA is basically, if you can think about it as like the polypeptides of amino acids, it is of course these nucleotides that form those long spiral that of course we know as DNA. Now, so how do we differentiate positive organisms from those that are negative? Well, the concept is actually very similar to that of the starch hydrolysis test. First, of course, you'll be looking for a clearing, and we can see that these 2 organisms here, compared with this one, do not have that distinct clearing around the colony itself. Now, this clearing forms because as this organism produces the enzyme called DNases, and it is sort of released into this surrounding area, those DNases will then break down the polymerized DNA into its simpler component known as nucleotides. And as I mentioned earlier, methyl green can only form a complex with DNA. And so when the DNA surrounding this colony is broken down into nucleotides, the methyl green can then no longer form this complex. And so you sort of see this light color forming around this colony, which indicates that that organism is positive for DNases. All right, and last but not least, the 3 core concepts that I want you guys to take away from this test. Number 1 being, of course, the functionality of the test itself, which is to look for organisms that can produce this hydrolytic enzyme known as DNase. Those DNases will then break down the main substrate known, which is polymerized DNA, and will then break them down into simple nucleotides. Of course, the methyl green indicator, which initially had formed a complex with the polymerized DNA, can no longer do that because it cannot form that complex with just nucleotides themselves. And so, of course, organisms that produce DNases will form that very distinct clearing around its colony. And finally, the application for this test, it is used to distinguish Serratia from Enterobacter and Staphylococcus aureus from other Staphylococcus species. So just like in the previous test, perhaps if you were in a situation where you knew you were dealing with potential Staphylococcus species, this is a great test to use to see whether or not you potentially may have Staphylococcus aureus or some other Staphylococcus species such as Epidermidis. Alright guys, that brings us to the end of our video. I hope you guys found this helpful. The next experiments are part of week 11, and I am working on those videos right now. And so, once again, if you like this video, please like it. Thank you so much, and I will see you guys next time.